Hello everyone, my name is Idowu Olawoye and I will be taking you on Module 3 Variant Lineage Identification, and with my colleagues John Juma, Joan Bernard and Nicholas Bosa will be also taking this course with me. So these are the outlines and the scope that we're going to be covering today. We'll be looking at what is a lineage, what is a variant, what are variant or lineages of concern, and examples of SARS-CoV-2 lineages. So in a context, as we all know, pathogens evolve continuously in the ecosystem, and this can happen within a host, that is when an individual is infected with a virus. This virus replicates, and during the replication cycle the virus would undergo certain mutations. However, these mutations are rare mutations, which is to say the differences between the viral copy in this individual are all the same except for certain differences that are hard to pick up. And also we can have variation at a population level, which is when a person infects a group of people. For example, in a conference and someone starts a super spreader event, when this person disperses this virus within that population, the infected individuals would have the same copy of the person with site infection. However, they would also have certain mutations as this virus replicates in this new host, which is what we call between host evolution. And that's within the scope of what we'll be covering today. When you have population and then you're sampling from that population, you're trying to distinguish what variants and what mutations are within that population. So why should we worry about mutations? Let's take an example of the mutation in SARS-CoV-2 genome like we have in the slide. And you can see the slide show the diversity that can be observed at each genome position of the Omicron BA2 strain. And if you notice, you'd see some position of the genome or some genes are much more variable than others. Take for example, look at the spike protein. You'd see this has lots of mutations compared to the ORF1A and the ORF1B genes. And this is something we scientists always try to look out for when looking for mutations. You want to understand where are the hotspot genes or where the mutations occur frequently, and that's where we start to worry. What are the implications of these mutations? But the mutations that involve or that occur within a population of virus, some might not really cause any harm to the human. However, some might confer more virulent properties, and that's why it's important to study the implication of these mutations. And to study the importance of the single mutations or group of mutation, we can associate them with the dynamics of the lineages that they are associated with. That is to say, if you notice the mutation is present in a group of population where that group of population are having severe symptoms compared to a group of the population that are asymptomatic and do not have that mutation, for example, that's also where you can start to link phenotypes with genotypes in the genome. Now let's define some of this term we are mentioning, or what we're talking about. So a lineage is a phylogenetically defined group of sequences that encompasses the ancestors and the descendants. And on a phylogenetic tree on the left, you can see there are two parts, one highlighted in red and the other in blue. So this corresponds to two lineages. You have the lineage A and the lineage 2, and the lineage B rather. And however, this lineage reflects the evolutionary relationship between the sequences in this tree. And if you look in detail, you'd see they all have a common ancestor. However, at some certain point, they broke out into two different lineages, and from there and that's based on certain mutations that define or segregate these groups. And down the line, you would see that they start to break down into distinct clades or distinct lineages, where you'd see you'd see the internet node break out into a new cluster of sequences, and these are all caused by mutations in the sequences. And these mutations on the branch leading to the node defining the blue lineage are all shared between the sequences in that group, which is to say all the sequences in this group would all have a mutation that they all share together, which is at the node where they broke out. And then if you then narrow down into a clade, those group of sequences in that clade will have defining mutations that are only specific to them, that you would not find in other clades. And the way lineages are defined on a phylogenetic tree varies depending on the pathogen. For SARS-CoV-2, there are several ways to define lineages, and there are different nomenclatures used, or naming schemes used in defining these lineages, but they are all used to track the pathogens. That is, the names given to them are easily used to pinpoint what lineage is circulating within the community, and therefore it helps to track the spread of that pathogen. For example, if you have the A1.1 causing an outbreak in the locality, we can then track the spread of this lineage by identifying what mutations are present in that group of viruses, and then also give it a name that helps us understand the evolutionary relationship, or how it evolved to that position. And that's how we define lineages. Now that we know what a lineage is, let's see what a variant is. So the term variant in itself means genetically distinct due to mutation or set of mutations, and we are usually concerned about a frequency of a variant with collection of mutations that have evidence of functional importance at a population level. Which is, if you find a mutation at a specific protein and you see that mutation is increasing in number during an outbreak, what we scientists are really worried or concerned about is what evidence in the lab do we have that shows that this mutation is of functional importance. Does it make the virus much more deadly? Does the virus evade vaccines, or does it escape immune response at a population level? Which is to say, when you say a mutation like increase in number, it is definitely conferring, or obviously conferring, some fitness to the virus, which helps it to spread faster. And that's why we want to know what are the implications of these mutations. So just as pathogens constantly evolve within a host and at a population level, it would continue to detect variants, because viruses continue to evolve and mutate as they replicate. And within the population, when someone infects another person, the virus will mutate, and then we will just continue to detect variants, because the virus continues to be spread between the population. For example, the SARS-CoV-2 virus acquire about 2 mutations per month at population level. However, some other pathogens evolve at a much lower rate. Take, for example, tuberculosis. It evolved at a much lower rate than SARS-CoV-2. I think about a mutation in 2 years or thereabout. When a mutation or group of mutations corresponds to variants that occur at the sites in the genome, it may be important for the function of the pathogen. Just like I said, it might give the virus more fitness to spread in the population. And it is interesting, or it is of importance, to track the frequency of this lineage associated with that variant in the overall population to understand if it may be under selection. So, how do we know if a variant is something we should be concerned about? How do we know if it's a variant of concern? Do the public need to be worried? Do the scientists or the doctors need to be on the edge to receive more patients in the hospital? We don't know. But how do we prepare ourselves for the next outbreak? So, like I said, all pathogens evolve continuously. And for SARS-CoV-2 viruses, as they acquire mutations, it affects how it shapes the pandemic, and such as like increased transmission. We've seen that with certain variants. Some could confer immune escape through the mutations that they harbor. Some can even cause vaccine breakthrough infections, which means after you've been vaccinated, you can still be infected by this virus. And some viruses might also cause severity of symptoms and all of these are usually termed variants of concern. This means when you have a virus in the population, and this virus or the strain of this virus, due to the mutations it has accumulated, is causing one of these characteristics. Maybe people are having more severe symptoms, or people who have been vaccinated before then being infected by this new variant, or maybe the intervention that's been placed is not containing the spread of the virus, and that's when we know this is a variant of concern, because it's of public health importance. And when does this happen? When do we see the rise of infections, or how do we know we have a variant that's emerging and this variant is going to be a variant of concern? So in many cases, variants co-circulate amongst themselves. You can have a variant, maybe variant A or lineage A, coexisting with lineage B. However, there will be different proportions at which they cohabit. Let's take, for example, the Alpha variant that was first detected in the UK in 2020. Scientists noticed that there was a surge of infections, and these infections were all caused by a certain lineage when they did genome sequencing. And from genome sequencing analysis, they noticed that this bunch of sequences have certain mutations that are shared between that cluster. However, that was not the only lineage that was circulating at the point in time, but it had more frequency than the other variants. And through surveillance, they were able to see the proportion of the frequency of how these variants dominated other lineages or other variants. And that's how you monitor when you have variants circulating and dominating other variants. If you look at this chart that I have in the screen here, you'd notice at some certain point in time, a lineage would dominate other lineages. For example, if we look here at January 2022, you have the BA1 dominating, and at a certain point it died out and another variant took over, which is the BA5. And at a certain point in time, the BA5 started to die
down again, and then a different variant starts to take over. That's the BA5.2, which is a subvariant of the initial variant. And in the time course of things, during space and time, you would find that a variant would come and go, and another would come and take over and then die down as well. And how do we track all of these events? And this is done through genomic surveillance, when you're sequencing viruses from patients and you're trying to monitor the frequency of this variant as it's spread within the population. So should we always be concerned when you find a variant increase in frequency? Yes, we should be worried, because that means that variant has gained some fitness and it's spreading much more in the population. So if a variant spreads within the population, it means it has evolved to transmit better, and thus it has selective advantage over the other circulating variants. So yes, we should be concerned if we see an increase in frequency, but mostly we should also investigate to see if this is due to higher transmissibility, because sometimes you might have some bias in sample selection. So what do we know as variants of interest? So a SARS-CoV-2 variant with genetic changes, also known or that are known or predicted to cause increased transmission, immune escape, or disease severity, can be termed a variant of interest. What that means is when you notice a variant in an outbreak, and that outbreak probably has a distinct future when you do genome sequencing. Maybe in the spike gene you have a mutation that you've carried out tests or experimented in a lab, and it shows in animal model that this virus spreads within the population. However, you haven't seen the real implication in the real life pandemic or outbreak, which is to say maybe there are not increased hospitalization, or there are no increased mortality, or there's no vaccine breakthrough infections, but the virus actually has mutations that can be proven in the lab setting that it is a variant or mutation of concern. Then you term this variant a variant of interest. We've had a couple of these in the pandemic, such as the E-type variant, which is also known as B1525, and we've also had a couple of these in different localities as well, where you'd have an outbreak and then that variant would dominate for a couple of months and then die out. So we have an example of SARS-CoV-2 lineages here. You have the B117, also known as the alpha variant, the B1351, also known as the beta variant, the B1525, also known as the eta variant. This is a variant of interest, not a variant of concern. And then we also have the B11529, also known as the Omicron variant, which is a variant of concern. And then we also have the B16172, also known as the Delta variant. So how can we identify variants? So SARS-CoV-2 variants can be identified by using classification software such as PANGOlin or NextClade. So this is a software that takes your sequences or your genomes and compares them against the SARS-CoV-2 reference genome and other publicly sequenced genomes, and reports what mutations are present in the query sequence and the genome position. Which is to say, as more genomes are being sequenced and are being shared openly, we have a database of SARS-CoV-2 genomes and the associated variants of these genomes. So when you put your sample through one of the software, it would compare your sample through a database and reference sequence also, and then see where your sample closely matches to that database, and then classifies it as parts of that lineage or variant if it identifies one. This is an example, or this is the screen shot of PANGOlin. So PANGOlin is an acronym for phylogenetic assignment of named global outbreak lineages, and it is a web application, and what it lets you do, it allows you to upload a FASTA file onto the website, and then it starts the analysis and tells you what lineage your sample is. Aside running this on the web application, you can also download this and run it from the command line. So when you run your samples on PANGOlin, this is what the result would look like. You would have the FASTA file which you've uploaded. This is a multi-FASTA file, which is a file that contains several files, 6 in this case, and these are the individual 6 files in this collection of files. And if you look on the file right here, you'd see the lineage of these individual files. This is BA5.2, which is an Omicron variant. This is also an Omicron variant, and you have BL.1, which is also an Omicron variant. And then you have, these are all Omicron variants, but different sub-lineages of the Omicron variant. And this will tell you if there are assignments conflict, which means how sure is the software telling you that this is the right lineage. And if you notice, the majority of them have 0.0, which means there is no assignment conflict here, except for this, which has 0.5, which means that it's either the sequence is not good quality, or there are some errors in the consensus sequence. So the software cannot accurately identify it to the correct lineage, but this is the closest that it can assign it to. And once you click on the tick sign on one of the samples, it then gives you extra information about the lineage. For example, the Japan sequence, it's telling us that this is an Omicron lineage, and the Scorpio support, which is 0.98, which is, it's very certain this is Omicron. 98% is really good. And these are the just a note about the Scorpio core, which is how many alleles it's found in that sequence that are associated with that variant. And also, if you click on one of these buttons, it takes you to a page where you'd find out more information about that variant or that lineage. So alternatively, you can also download your result as a CSV file, which is containing the lineages in your FASTA file. And this is just the same information you have in this previous screen, but in a CSV file which you can display or open with a spreadsheet software, and you can use that to generate a report of some sort. So additionally, if you click one of these buttons, like I said earlier, it can also give you more information about the lineage if you would like to know more. So for example, the BA5.2 is a parent lineage. The parent lineage is BA5, which means that's the ancestor. And this is the actual name or the alias, which is B11.529.5.2. And these are the locations it can be predominantly found. And then the countries with highest frequency of this lineage, and the first date the lineage was detected, and how many sequences in the database, which is GISAID, have been designated to this lineage, and how many have been assigned. So this is NextClade. So NextClade is a software that assigns lineages to your sample, just like PANGOlin does. But NextClade also has extra functions, which is it does quality control, gives you a quality score of your samples, and also gives information about the mutations, what positions they are in your genome, and informations that can be of good importance to a bioinformatician like yourself. So NextClade can also be accessed on the web server or the web application, and you can also download it to your computer and run it through your command line. And all you have to do is select the pathogen you're working with, which is SARS-CoV-2 in this case, and then you can simply drag and drop your multi-FASTA file or browse your computer using this button here. And once you do that, the software will execute the program and try to classify your samples to the right lineage. So these are the same 6 samples, the same 6 genomes we run through PANGOlin, and this will tell you the clade. So NextClade has a naming nomenclature it uses, and this is quite different from what PANGOlin uses, but they are the same. It's just one is just more easier to read. For example, in NextClade you have 22B, which is the same as BA 5.2, and you have 22D, which is this. And if you notice, 22D can be multiple lineages, but it's a big clade. For example, 22D contains BL1 and BA2.75.2, and the same thing 22B contains BA.5.2 and BF7. So PANGOlin is much more distinct, or much more, it classifies the lineages to a very distinct level. However, NextClade can group different or multiple lineages to a big cluster, but they're all just like what I showed initially in that phylogenetic tree, where you have an ancestor and you have the descendants, where the ancestor we have mutations that you would all find in a descendant, but the descendants would have distinct mutations that groups them in a cluster, and that's what NextClade does as well. So in your NextClade result, you'd find things like number of mutations that are in your samples, and you also find numbers of ends, which are also missing information that you find in your genome, where you cannot assemble a full genome. And you also have statistics on the coverage of your genomes. How does the genome span the reference genome? And you have gaps, insertions, frame shifts, and you also have this graphics that shows where your mutations are in the genome. And also with NextClade, you can download your results as a CSV or TSV file, and then you can display that on your spreadsheet software. And the also nice thing about NextClade is that if you hover your mouse around the results on your computer screen, it's going to show you information about the mutations. For example, this sample has 85 substitution mutations related to the reference sequence, and you can see the positions of all these mutations. You see at position 241 changes T, and so on and so forth. And this also previews the amino acid substitution relative to the reference sequence, which means in the spike protein at position 19, a T changes to an I, and so on and so forth. So you can, all of this information are going to be contained in that CSV or TSV file that you download to your computer. So that is all I have for us today on lineage and variant detection for SARS-CoV-2. You can always go to the website of this software, PANGOlin and NextClade, to
find out more information on how the software works, and during the hands-on, you'd be able to download this software via command line, launch the software and run a test data on this software. And if you have any questions, feel free to reach out to any of my team on Slack, and we'll be happy to help. Thank you very much for listening.